Hi friends, welcome back to my channel. Today we will be dealing with the second part of antimicrobial drugs. Topics which we will cover today are antibacterial drugs, antiviral drugs and antifungal drugs along with their mode of action. Let's start with the antibacterial drugs. So mainly it can be divided into cell wall inhibitors, protein synthesis inhibitors, metabolic inhibitors and nucleic acid inhibitors. For GATE exam, it's very important to study the example of each of these drugs. It's not easy to remember all these examples, right? So we have to remember it through some codes. For example, in case of cell wall synthesis inhibitors, we have penicillin, cephalosporin, vancomycin and teicoplanin. So these are the important examples of cell wall synthesis inhibitors. Here we can make the code like PC and TV. Both are electronic devices. So you can remember it like that, PC and TV. In case of protein synthesis inhibitors, there is aminoglycosides, tetracycline, macrolide and chloramphenicol. You can remember it as ATM and you have to feed C into that additionally. Okay. Next is metabolic inhibitors. They are sulfonamides, trimethoprim, dapsone, isoniazid. You can remember it by STD along with I. Next one is nucleic acid inhibitors. So nucleic acid synthesis inhibitors are quinolones and rifampin. Next, let's have a look at the mode of action of antibacterial drugs by considering the example as penicillin. We all know that penicillin is derived from 6-aminopenicillinic acid. So from 6-aminopenicillinic acid, we will derive penicillin, and penicillin is having its bioactivity because of the beta-lactam ring, which is attached to the amino group of the penicillin. And why this penicillin is having this antibacterial activity? It is because its structure is very much similar to D-alanine D-alanine, which is present in the peptidoglycan membrane of the bacteria. What will happen? The penicillin will block it will prevent the transpeptidation reaction, which is very essential for the formation of the peptidoglycan crosslinks. The NAM and NAG links will not be formed without the transpeptidation reaction. What will happen then? The cell wall synthesis will not happen in bacteria. If there is no cell wall, then what will happen? Bacteria will die. Okay, so what the bacteria will do, they are very clever. They will start developing some resistance, or they will try to avoid this penicillin. They will produce an enzyme called penicillinase, which will act on the beta-lactam, and it causes the hydrolyzation of some bonds in beta-lactam. Thereby, it will prevent the activity of the penicillin. And you have to remember one thing also, that this is not only the mechanism used by penicillin. There are several mechanisms by which penicillin will kill the bacteria. One among that is the binding with penicillin binding proteins. It may also destroy the bacteria by activating their own autolytic enzymes. However, this penicillin may kill bacteria even in the absence of this autolysins or murine hydrolysis. Lysis could occur after bacterial viability has already been lost. So penicillin may stimulate protein called bacterial holing. Okay, so penicillin will result in the stimulation of bacterial holines to form hole or lesion in the plasma membrane of the bacteria. So this result in the formation of holes in the plasma membrane of the bacteria, leading to directly to the membrane leakage and finally to the death. And other one is murine hydrolysis. It could also move to the holes and disrupt the peptidoglycan and lyse the cell. So this is how cell wall synthesis inhibiting drugs will act on the bacteria. Now let's see the action of protein synthesis inhibiting drugs, which are the aminoglycosides, tetracyclines, macrolides and chloramphenicol. So how is the action of aminoglycosides? Aminoglycosides bind to the 30S subunit of the ribosome. Likewise, tetracycline also bind to the 30S subunit, where macrolides and chloramphenicol bind to the 50S subunit. You have to remember this. Okay. First, let's see the action of aminoglycosides. Aminoglycoside bind to the 30th subunit of the ribosome and they leads to the tRNA mismatch. What happens if tRNA is mismatched? The protein mistranslation will occur. The mistranslated protein along with the correctly translated protein, they will move into the periplasm by the sec-dependent translocation system. So there is a translocation system in bacteria which is sec. Okay, so this through the sec-dependent translocation system, the corrected as well as the correctly translated protein as well as the mistranslated protein will reach the periplasm. Once in the periplasm, the mistranslated proteins are degraded, but not before some of them are inserted into the plasma membrane, activating the envelope stress response system. Okay. Finally, the increased oxygen radical produced here causes the cell death. The next one is tetracycline. Tetracycline is also acting similar to aminoglycoside. They will also bind to the 30S subunit of the ribosome and they will inhibit the binding of amino-acyl tRNA to a site of the ribosome. So what is action? There is different site like EPA. These are the sites of the ribosome. So when amino-acyl tRNA will come and bind to this A site, this tetracycline will prevent that. Okay, so that is the action of tetracycline. So the next one is macrolides. In case of macrolides, they will bind to the 50S subunit and that also in 23R RNA of the 50S subunit of the ribosome, 50S ribosome, and they will inhibit the elongation. Okay. Finally, coming to the chloramphenicol, they will also bind to the 50S subunit of the ribosome and they will inhibit the peptidyl transferase reaction. So this chloramphenicol, actually it has a very broad spectrum of activity, but unfortunately it is very toxic. Means it has high cytotoxicity. It can affect the human cell also. That is the problem. Okay. Now let's see the action of metabolic antagonist. The example of metabolic antagonist which we studied were sulfonamide and trimethoprim. So how is their action? The sulfonamide, which is structurally related to PABA. This PABA is para-aminobenzoic acid. This is an important factor for the synthesis of cofactor folic acid. What happens is when sulfonamide enters the bacteria, it will compete with the site where PABA binds. And what is the action of PABA? It is for the synthesis of folic acid. When sulfonamide is binding to that site, the synthesis of folic acid will not take place. So this bacteria will not produce any folic acid. So what happens if folic acid is not there? We all know that folic acid is a very important precursor of purines and pyrimidine. So if no folic acid, that means no purine and pyrimidine. Then there will not be any DNA replication. Thus the death of the bacteria. And another important factor is the sulfonamide is very specific because most of the bacteria, they do not have any ability to uptake this folic acid. They will synthesize folic acid by their own. So if that folic acid synthesis is blocked in the bacteria, they will not be able to uptake this folic acid from the surrounding environment also. In case of humans, humans will not produce folic acid inside their body. They're always uptaking it. So if any mechanism which will prevent the synthesis of folic acid or any sulfonamide drug is acting on our cell also, there will not be much toxicity happening to our body. Therefore, because of this specificity, sulfonamide is having a high therapeutic index. Next is nucleic acid synthesis inhibitors. So examples which we studied were quinolone and rifampin. Right. What quinolones will do is it will inhibit two enzymes, DNA gyrase and topoisomerase 2. So what is the function of DNA gyrase? It will help in the separation of strands. Okay, it will produce negative twist in the DNA and it will help in the strand separation. So what this strand separation is for? For the DNA replication, repair and chromosome separation. So when DNA gyrase is blocked or inhibited, what will happen? All this process will not happen. In case of topoisomerase 2, same thing. It will untangle the DNA during replication and it is blocked, so there will not be any DNA replication. So without DNA replication, there will not be any synthesis of nucleic acid, right? This is how they will perform their role. Next is antifungal drugs. Antifungal drugs are actually used against the fungal infection. It can be divided into superficial, subcutaneous and systemic. So in order to treat the superficial mycosis, we use miconazole, ketoconazole and clotrimazole. And to treat systemic infection, we use amphotericin B, 5-flucytosine and fluconazole. For subcutaneous infection, we can use the combination of both of these. Antiviral drugs are the drugs which act against the virus. So because of the virus enter the host cell and make use of the host cell enzyme and constituents, it was long thought that a drug that blocked the virus multiplication could be toxic for the host also. But scientists were able to successfully produce some of the drugs which inhibit the virus in different stage of their life cycle. So most of these antiviral drugs disrupt either critical stages in the virus life cycle or the synthesis of the virus specific nucleic acid. For example, this amantadine and rimantadine, they can be used to prevent the influenza A virus. And vidarabine, which will disrupt the activity of DNA polymerase. Next one is acyclovir. It is used in the treatment of herpes infection. Next is cidofovir. Cidofovir is active against many viruses. It is effective against many viruses. Example, parvovirus, adenovirus, and pox virus. Okay. And next is azidothymidine, which we all know. It is a anti-HIV drug. So these are some of the examples of antiviral drugs. So that's all for today. Hope you all understood the topic, and if you like this video, please like and share and subscribe my channel. Thank you.